we will kick off with the keynote lecture by Bill Aird, and he is going to help us see the endothelium.
All right, thank you. That's not a good start to be disoriented to time.
One more request is to use your mouse to indicate we are on videocast. So there are hundreds of people watching us on videocast, and the only way they can see what you are showing on the screen is by using the mouse to move around. So
Where is the mouse?
Yes.
Okay,
here.
thank you.
Would you like to take it?
Yep,
here?
great.
Okay.
All right. Well, thank you to the organizers for this invitation to speak today. Since this is the opening talk, I thought that rather than showing a lot of primary data, that I would take this opportunity to take some creative license and to tell a story to help set the mood and to at least begin a dialogue. And the story is about how, as scientists, we identify and choose to see objects that are worthy of scientific attention, in this case, the endothelium, and how that prism changes not only between individuals, but also over time, according to the prevailing methodologies and technologies of the day, to historical constraints, particularly subspecialization, and of course to prevailing therapeutic imperatives. So all vertebrates have a closed vascular system in which blood is maintained within the confines of blood vessels and is propelled around the body by the action of a central pump or the heart. Now, the very term system implies cohesion, interaction, actually in this case, continuity. But it's our natural tendency to break systems like this into its component parts, into bite-sized, manageable pieces that allow us to train our focus on one level or another. How and where we choose our lines of cleavage, however, very much depends upon our own individual idiosyncratic interests and biases. So the oldest classification system, which dates back to the discovery of the circulation in the 1600s and is favored by our pulmonary colleagues, separates the cardiovascular system into a pulmonary or lesser circulation and a systemic or greater circulation. Now, the importance of this schematic is that it emphasizes the role of the lung in the pulmonary circuit in revitalizing or reoxygenating blood as it makes its journey through the body. Another classification, which was introduced by Bichat in the early 1800s and found a staunch advocate in August Krogh in the early 1900s, separates the system into a propulsive organ and a series of distributing, exchange, and collecting pipes, the arteries, the capillaries, and the veins, respectively. This system recognizes the importance of the capillary bed as a structurally and functionally distinct entity. Now, cardiologists are necessarily focused more narrowly and intensely on the heart and its vascular supply, whereas our blood-brain barrier colleagues often refer to the rest of the circulation simply as the peripheral circulation. Now, another way to cut the system at its joints is to start peeling back the layers of the heart and the blood vessel wall. And in doing so, we find that the endothelium is the single entity that enters into the composition of the entire system. It's the common thread, the one piece that ties everything together. And viewed from this kind of delightfully endothelial-centric perspective, the endothelium is the fundamental vascular tissue, and the heart really is nothing more than a great muscular thickening around a portion of the system of endothelial tubes. Now, how we name and classify phenomena in nature is not a matter of semantics. It really matters. It influences how we think about and understand the natural world, what questions we ask and how we go about answering those questions. So in that spirit, I would like to submit that the endothelium, and by extension the capillaries, as I will show, should be viewed as a systemically relevant organ system, one that is teeming with life, every bit as active as any other organ system, and one that has largely untapped but enormous diagnostic and therapeutic potential. In fact, I would go so far as to say that the endothelium is an organizing principle in health and disease. Now, the goal of my talk today is not so much to convince you to share this point of view, I suspect some of you already do, but rather to ask the question, why don't more people see the endothelium in this light? So with that introduction, I'd like to focus our lens squarely on the endothelium. The endothelium is a spatially distributed organ system that reaches to almost all recesses of the human body. In an average-sized human being, it weighs about 1 kilogram. It covers a total surface area between 4,000 and 7,000 square meters, and it spans over 100,000 miles of blood vessels, the vast majority of which, the capillaries, are invisible to the naked eye. The endothelium is not an inert layer of nucleated cellophane as it was once thought to be, but is involved in many physiological functions, some of which are included on the slide. For example, control of barrier function, leukocyte trafficking, the maintenance of blood fluidity and the cessation of hemorrhage, the control of vasomotor tone, innate and acquired immunity, and of course new blood vessel formation. Endothelial cells secrete. They contract, they swell, they buckle. Some of them scavenge, others undergo transdifferentiation, and yet others are critical for organogenesis during development. Endothelial cell phenotypes are not uniform, but rather vary in structure and function in space and time, as well as in health and disease. And this gives rise to the phenomenon that we term in the vascular biology community endothelial cell heterogeneity. For example, endothelial cells that line most blood vessels, particularly capillaries, are extremely thin, whereas those that line the high endothelial venules of the lymph nodes have a cuboidal, plump, or tall morphology. Endothelial cells that line the straight segments of arteries but not veins are oriented along the longitudinal axis of blood flow. The endothelium of arteries and veins forms a continuous monolayer with its cells held together by tight zipper-like junctions. The endothelium of capillaries may, like their arterial and venous counterparts, be continuous, or they may be permeated by tiny holes or fenestrae. Or they may contain frank gaps within the endothelium and a poorly formed underlying basal lamina, the so-called discontinuous endothelium, depending upon the needs of the underlying tissue. For example, fenestrated endothelium is characteristic of those organs that are involved in a high degree of secretion and filtration. From a functional standpoint, endothelial cells display a remarkable division of labor. For example, endothelial cells that line arterioles are primarily responsible for mediating vasomotor tone, and they do so through the regulated release of a number of vasodilators and vasoconstrictors whose balance mediates the diameter of the blood vessel lumen and hence peripheral resistance. On the other side of the capillary bed, endothelial cells that line venules participate in the host innate immune response, promoting plasma leakage as well as leukocyte trafficking. This latter process in turn is mediated by the selectable site-specific upregulation of a number of adhesion molecules which create a Velcro-like surface that leads to rolling, firm adhesion and ultimately transmigration of white blood cells. Heterogeneity is particularly prominent at the level of capillaries where endothelial cells are adapted to the needs of the underlying tissue. From a molecular standpoint, endothelial cells from different sites of the vascular tree express different transcriptional profiles. In vivo phage display have uncovered site-specific repertoires of cell surface proteins or receptors that have been referred to as vascular zip codes. And this is a really nice example of metaphor, which has a very rich history and tradition in the field of vascular biology to describe what can't be seen with a corporeal eye, but rather with the mind's eye. So terms like cellophane, gatekeeper, vascular zip code comes to mind. And at the end of this talk, I will rely on the metaphor, very heavily on the metaphor of endothelial color palette. Now, the arteries and veins are the conduits or highways of the system, serving to deliver blood to and from various tissues of the body. In keeping with the conduit function, the walls of veins, and particularly arteries, are extremely thick, composed of multiple layers of smooth muscle cells as well as elastin that allow the vessel to withstand high systolic pressures and to provide elastic recoil during diastole. If the arteries and veins are the conduits of the system, the capillaries are the business end, or the marketplace, so to speak. This is where all the exchange takes place of nutrients and gases between blood and underlying tissue. Since the primary purpose of the circulation is to maintain and promote such exchange, then viewed from this perspective, the heart, the arteries, and veins are essentially subservient to the functions of exchange carried out in the capillaries. To drive this point home even further, consider that arteries and veins require their own capillary supply, the vasa vasorum, for nourishment as well as oxygenation of their walls. Now, based on these and other considerations, August Krogh, who was one of the great physiologists at the turn of the 20th century, concluded that it will be difficult to challenge the proposition that the capillaries constitute the most important part of the whole circulatory system. Now, in keeping with Fick's law of diffusion, Capillaries comprise the vast majority of the surface area of the vascular tree. Moreover, they're extraordinarily thin. They're essentially three-dimensional tubes of endothelium that are telescoped within a sheath or a sleeve of basement membrane and then wrapped to a variable degree or cradled by highly branching pericytes, which have a contractile function which provides stability and integrity to the capillary wall and which represent a potentially important niche for mesenchymal stem cells. Now, a typical capillary is just wide enough to accommodate a single red cell, which march in single file, one by one, from arteriolar to venular end, with each cell in a position of reciprocal barter with the underlying endothelium, which further enhances the diffusional capacity of the system. However, there's some interesting exceptions to the canonical arrangement of the microvascular unit. As an endothelial biologist, these are really interesting because each one of these variations entails underlying differences in endothelial cell phenotypes. So for starters, there are two vascular beds or circulations in the body in which the oxygen gradient in capillaries is reversed. That is the pulmonary circulation and the placental circulation. So in these cases, endothelial cells on the arteriolar end of the capillary are exposed to high rates of flow of relatively deoxygenated blood. Not all capillaries are perfused all the time. August Krogh won his Nobel Prize in 1920 for the discovery of a reserve of unperfused capillaries in skeletal muscle, which then become recruited when there are increased metabolic demands, as occurs, for example, with exercise or contraction. Now, Krogh believed that this phenomenon was mediated at the level of the single capillary. However, today it's more widely held that capillary perfusion is mediated or regulated primarily by arterioles. There's some interesting exceptions, including the vasa recta of the kidney, and based on some really elegant recent studies on the blood-brain barrier in capillaries of the brain. Now, when those capillaries that are perfused, flow is generally pulsatile. So, for example, in the dermal capillaries, pulse pressure has been measured between 0.5 and 12 millimeters of mercury. A really cool exception, however, are the cochlear capillaries in the inner ear, which are fed by highly convoluted and tortuous arterioles that are believed to dampen the pulse wave, converting a pulsatile flow into a more linear stream. And without this adaptation, we would be faced with having to listen to our heartbeat day in and day out, a consequence that would not only drive us crazy, but would also mask important auditory signals. Not all arteries are connected to veins by capillaries. In some cases, blood is shunted from the arterial to venous side through AV and anastomoses. These are normally seen in the skin, particularly in the fingers and the toes, where they serve as heat exchangers, diverting up to 85% of the blood flow to the dermis. These are also seen in pathology where they're called AV malformations, for example, in certain hereditary states, as well as in tumors. Not all capillaries are teeny tiny hair-like tubes. In some cases, arterioles empty their blood into lakes or sinuses. So we're all very familiar with the sinusoids of the liver, the spleen, and the bone marrow. A more striking example is the placenta. In this case, in humans, a series of about 100 to 120 open-ended spiral arteries pump blood into the intervillous space, which, in a nice case of vascular mimicry, is lined not by endothelial cells, but rather by fetal trophoblasts. Maternal blood then bathes chorionic villi, which contain capillaries from the fetus, allowing for exchange to take place. These capillaries are fed by the umbilical artery and are drained by the umbilical vein. Maternal blood then returns to the system through the uterine vein. Now, normally during pregnancy, fetal trophoblasts invade the spiral arteries, remodeling it into a high-capacitance, low-resistance vessel. In preeclampsia, remodeling and invasion by trophoblasts is defective, leading to hypoxia of the placenta, and the release of circulating factors then spill over into the maternal circulation and have downstream effects. Perhaps the most extreme, if not dynamic, situation is that of the male erection. So in a response to appropriate neurophysiological stimuli, blood rushes through dilated penile arteries into the corpus cavernosum, which is essentially one enormous spongy sinus. At the same time, there is compression of venous outflow, leading to complete obstruction, rapid filling, expansion, and engorgement of the sinusoidal spaces, increased pressure, and ultimately rigidity. Now, all organismal pleasures aside, consider this from the standpoint of the endothelial cell. So at the drop of a hat, with absolutely no warning, endothelial cells are called into action, jolted from their usual state of tranquility and serenity to cope with this massive infusion of blood which causes enormous pressures, up to 300 millimeters of mercury in human, up to 600 millimeters of mercury in the bull. And because that blood can't escape, rapid stagnation as well as deoxygenation. So it's no wonder that priapism, which is essentially a compartment syndrome, is a medical emergency. Viagra works as a phosphodiesterase inhibitor by increasing cyclic GMP levels within the arteriolar smooth muscle cells that allows increased flow of blood into this sinus space. Now, not all capillaries connect arteries to veins. For example, the hepatic sinusoids are perfused not only by the hepatic artery, but also by the portal vein, which carries deoxygenated blood, and particularly in the postprandial period, is full of partially digested food. In the kidney, glomerular capillaries connect two arterioles, the afferent and efferent, which then is distributed into yet another capillary bed, the peritubular capillaries. Now, the glomerular capillary is an ultrafiltration unit. There's very little uptake of oxygen and nutrients, whereas the peritubular capillary is more of a traditional nutrient bed. Now, in some cases, the efferent arteriole dives into the depths of the inner medulla. In this kind of no man's land, endothelial cells have to balance the competing needs not only of delivering oxygen as well as nutrients to the tissue of the inner medulla, but also to function in countercurrent exchange and to adapt to and survive in extremes of environment. For example, oxygenation levels PO2 of less than 10, a hyperosmolar and a hyperkalemic environment. Now, all of these variations are important reminders that while the primary purpose of the circulation is to transport substances, they have a number of ancillary functions. Heat regulation, I've mentioned, hydraulic force generation, ultrafiltration, and finally countercurrent exchange. I'm going to switch gears now from the level of the capillary to the level of its most important constituent, the endothelial cell. So here's a recent PubMed search in which I entered the terms endothelium and endothelial cells that yielded a total of 155,000 and 140,000 articles respectively. This amounts to an enormous expenditure of resources, not to mention sweat and toil on the part of largely basic scientists, but to some extent translational researchers. Collectively, the data provide compelling evidence that endothelial cells are involved in most, if not all, human diseases, either as a primary determinant of pathophysiology or as a victim of collateral damage. However, if you then peruse the table of contents or the index of the common or popular medical textbooks or on-call references, one finds little reference to these terms. And one of the more striking examples is Scientific American Medicine, which publishes a hard copy of about 100 pages of index with over 10,000 entries, not a single mention of endothelium or endothelial cells. I think it's fair to say that very few clinicians are attuned to the health of this cell layer. There is no syncope, diarrhea, jaundice, or bleeding equivalent for the endothelium. There's no endothelial box that we check off as we move through the review of systems. And the endothelium is not amenable to the traditional physical diagnostic maneuvers of inspection, palpation, percussion, and auscultation. While renal function is readily assayed by BUN and creatinine, liver function by bilirubin and the transaminases, hematological function by the complete blood count, the endothelium continues to defy convenient diagnostic interrogation. So this amounts to what I find and feel is the most striking example of a bench-to-bedside gap in biomedicine today. How is it that we can go from over 1 million pages of published literature to a virtual blackout at the bedside? Well, there's several considerations, 3 of which I'd like to touch on today. One relates to the hidden enigmatic nature of the endothelium. The endothelium flies under the clinical radar screen. It rarely shows its hand, at least in ways that we as clinicians are trained to detect. So while, like blood cells, circulating blood cells, the endothelium is distributed throughout the entire body, the endothelium is tightly tethered to the blood vessel wall. Therefore, it's inaccessible for diagnostic purposes. Moreover, unlike solid organs like the lung, the liver, the heart, the kidney, the endothelium is not well circumscribed. And in fact, its diffuseness and paper-like thinness makes it virtually impossible to image using conventional diagnostic techniques. While there has been some progress over recent years in developing novel diagnostic technologies, for example, measurement of circulating biomarkers, the enumeration and phenotyping of circulating endothelial cells and blood outgrowth endothelial cells, and more recently molecular imaging, these are all investigational and have yet to have a major impact on clinical care. Well, the second consideration is our tendency to think big, that is to focus on the large vessels, and to do so in ways that compartmentalize the body into organ-specific disciplines. So when we throw out the term vascular, whether in the context of medicine, surgery, biology, or research, most of us do think about arteries and veins. That is the sites of atherosclerosis and deep venous thrombosis, respectively. And for good reason. After all, acute myocardial infarction or coronary heart disease is the leading cause of mortality in the Western world. But it's noteworthy that we are focusing on just a few, albeit critical, inches of the vascular tree, really just a quantum dot on the surface area map relative to the vast expanses of microvessels that permeate every organ. And I think this is a really important take-home message from this talk, and I think from other talks that we'll hear in the next 2 days, and that is that an understanding of the structure and function of these microvascular beds holds important clues to the understanding the underlying pathophysiology and therapy of virtually every other organic disease state that contributes to this mortality pie chart. This idea is not new. So consider, for example, this prophetic statement that was written 130 years ago. Capillaries are the true markets of exchange. Hence the paramount importance of these vessels in the maintenance of life and health. Hence also the direct practical utility of knowing their minute anatomy and physiological dignity. Every practitioner of medicine will see the important relation this branch of histology holds to pathology and therefore to therapeutics. Now, 30 years later, August Krogh lamented that it's a little strange that the capillaries have been neglected in an extraordinary degree. Today, every subspecialty diagnoses and treats its own share of microvascular disorders, or at least those diseases that have a significant microvascular component. But they're not always recognized as such. There's nothing about these disease names or the way we think about them that shouts out capillaries or endothelium. Rather than constituting an integrated system, capillaries from different organs fall under the purview of subspecialties whose frame of reference is not the vasculature per se, but rather the physiology and pathophysiology of the organ in which the capillaries happen to reside. Now, this tendency to think about large vessels is deeply rooted in history. So the Greeks in the classical age and the Hellenistic age viewed the arteries and veins as largely uncoupled systems. And in retrospect, it's easy to understand why. They couldn't see the invisible capillaries. Rather, what they saw was a series of thick-walled, deeply situated, pulsating vessels that carried red-colored blood, and a separate network of thin-walled vessels that lay both in superficial and deep tissue planes, didn't pulsate, and carried a darker color blood. So according to Galen, blood is made in the liver by food that is brought in by the portal vein and is then distributed through the various veins of the body, where it replaces tissue that is lost by invisible emanation. Some, but not all of this blood enters the right ventricle, from where it goes through the pulmonary artery to feed the lungs, or through invisible pores within the interventricular septum, where the blood mixes with air that is brought in from the pulmonary vein. This air-blood mixture, or vital spirit, is then distributed to the various organs of the body by way of the arteries, where it provides motion and vitality and heat. These are open-ended, one-way distribution systems in which blood and air simply diffuse at the level of the tissue. Blood is not seen to circulate, but rather to ebb and flow like the tide, and the heart is not a propulsive pump. It's a furnace that provides innate heat to the body. Movement of blood and air within the arteries and veins is mediated by attractive forces that are exerted by hungry tissues. Now, following Galen's death, science and medicine moved into the monasteries, where both of these disciplines kind of languished under the ruling thumb of theology for the next 1,500 years. Galen's work fit well with Christian doctrine. In fact, his greatest work on the use of body parts is one long argument from design. To study medicine was to study Galen. The goal was not to investigate or discover, but rather to collect, to collate, to translate, to study, and to teach the work of the ancients. All of this would change in 1628 when William Harvey, an English physician, published his short book with a long title, 72 pages long, what's considered now to be one of the greatest works in the annals of biomedical history. Using a series of elegant physiological studies that represented more of a methodological rather than a technological advance. For example, the crude quantitation of cardiac output and the strategic use of tourniquets. William Harvey discovered that that arteries and veins are functionally, if not structurally, connected, and that blood is conserved rather than consumed. Once this was established as fact, which would take some years, just before Harvey's death, this would change the worldview of the human body and would lay the foundation for all of modern physiology and medicine. Now, like his predecessors, Harvey's could not see the invisible capillaries. He surmised they might exist, but he actually preferred the notion that blood percolated through the tissues, going from the artery to the vein. After all, he thought, how else could nutrients possibly get out from the blood vessel into the tissues? The discovery of the capillaries would wait for a technological advance, and that was the introduction of the light microscope. So in 1661, 4 years after Harvey's death, Marcello Malpighi, who was a professor of anatomy at the University of Bologna, peered down the eyepiece of his compound double convex lens looking at fresh preparations of frog lung and found that blood was not poured out into air spaces, but was always driven through tiny tubules. And then he went on to look at other tissues and other organs and found the same pattern virtually everywhere. Now, what's surprising by today's standards is that the following 200 years was characterized and in many ways paralyzed by a lively debate as to the entity of these structures. Were they simply canals that were drilled into tissue, or did they have distinct walls? And this question would not be answered until yet another technological advance, a really simple one in this case, and that was the introduction of silver nitrate staining, which is an intravital stain that paints the borders between endothelial cells, converting it from a bland, homogeneous, sort of glistening and moist membrane into a pavement of tiles with a cobblestone pattern. And this was the original paper by von Recklinghausen in 1860. This isn't an abstract. This is a paper, 8 lines long, no references, no figures. These were the good old days. Now, 5 years later, Wilhelm Hess decided to name the endothelium, to differentiate it from other types of epithelium. This was a major development because it said that the endothelium is now a thing. It is an object that is worthy of investigation in its own right. A very, very important development in the field of endothelial biology. Now, in the late 1800s, the use of intravital microscopy uncovered an important link between inflammation and permeability and leukocyte transmigration. This was the heyday of the microvessels, or the golden age of the capillary, and for good reason in retrospect, and that is, in this pre-antibiotic era, infectious disease, both the acute and chronic formed, reaped more havoc on the population than any other disease. And of course, infectious disease targets the microvasculature. In fact, atherosclerosis was not recognized as a disease, let alone a public health concern, until well into the 1900s. But once focus was changed, attention was altered to the large vessels, there was no turning back. And it really is not uncommon to see the cardiovascular scheme depicted as it is on the left-hand side of this slide. Not only does this vastly underestimate the breadth and scope of microvessels in the body, but it dangerously approaches a galenic view of the circulation. Now, the next major advance in the field of endothelial biology was the introduction of the electron microscope. This allowed investigators to break through the resolution barrier of light microscopy, converting the cytoplasm from kind of a bland bag into a rich, compartmentalized, three-dimensional universe of structurally and functionally distinct organelles. This early work was done by George Palade, who won a Nobel Prize for his discoveries. Now, as wonderful as these discoveries were, truth be told, there was nothing that was particularly unique about the endothelial cell, except perhaps the Weibel-Palade body. In other words, endothelial identity in the full sense of the word was not to be found at an ultrastructural level. At the end of the day, no matter how good the resolution or high the power, no matter how good the fixation and the staining or quality of the cutting life, we're still looking down the lens at devitalized dead tissue, and there's only so much information that can be obtained from these types of cellular autopsy. So in this graph, I've plotted the number of publications related to endothelium and endothelial cells on the y-axis relative to the year that they were published on the x-axis. Now, you'll notice smattering of publications in the 1950s and 1960s, hardly noticeable at this scale. And these are largely related to some seminal physiological experiments, as well as basic histology, cytology, and electron microscopy. Only so many stories you can tell. Something then happened in the early 1700s that changed the landscape. And this was the first isolation and culture of endothelial cells from humans, originally from umbilical veins from discarded placentas, described by two independent groups, Eric Jaffe and Michael Giambroni. Now investigators had a powerful means to dissect, to study highly purified populations of living cells under tightly controlled conditions. And this led to an exponential increase in the number of publications and enormous advances in our understanding of endothelial biology. However, it can be argued that these successes came at a price, that there were diminishing and are diminishing returns. The reason being that cell culture encouraged us to think about the endothelium as a kind of a monopoly of identical cells in a culture dish, rather than as an emergent consortium with properties, emergent properties whose whole is greater than the sum of the parts. And that brings me to the last consideration in explaining the bench to bedside gap, and that is the complexity of the endothelium. The endothelial cell is like a chameleon. It marches to the tune of the microenvironment. In fact, so tightly coupled is it to the microenvironment that when it's removed from its native tissue, it's like a fish out of water. It undergoes phenotypic drift and becomes a shadow of its former self, and as a result, we have to interpret in vitro-based assays with extreme caution. I like to think about each one of our trillions of endothelial cells as a miniature adaptive input-output device. The input arises from the extracellular environment, either the luminal side, the abluminal side, neighboring endothelial cells, and consists of any number of biochemical and biophysical signals. The output is the phenotype of the cell, so this depends upon the level of investigation. Single cells may undergo a change in calcium flux, a change in shape, protein expression, mRNA expression. Single endothelial cells may migrate, proliferate, undergo programmed cell death. Monolayers of endothelial cells, for example, in a tissue culture plate, express barrier properties and can be assayed for leukocyte transmigration. And yet other properties are apparent only at higher levels of organization, for example, at the level of the blood vessel, the organ, or the whole organism. And examples of these properties might include endothelial contribution to vasomotor tone or fibrin deposition, perhaps angiogenesis. These are properties that can't precisely be assayed for using in vitro conditions. Now, input is coupled to output by a nonlinear array of signaling pathways that typically begin at the cell surface by way of a receptor and end at the level of transcription or post-transcriptional control. Many endothelial biologists, and I include myself in this group, spend a lot of our career with our heads stuck in the sand trying to understand basic mechanisms of signal transduction. But if we step back and take a 30,000-foot view, what we'll realize is that the input varies across the vascular tree from head to toe. For example, endothelial cells in the blood-brain barrier are exposed to a marinade of astroglial and neuronal-derived factors that are essential for maintenance of barrier function. Whereas endothelial cells that line capillaries of the heart are exposed to specialized regional forces that are associated with the cardiac cycle, as well as paracrine mediators that are secreted by neighboring cardiomyocytes. Since input varies across the vascular tree from head to toe, and insofar as the endothelial cell is capable of transducing a signal, then the output has to vary from one side of the vascular tree to the next. In fact, if we could somehow color code the endothelium, that is, assign it a shade of color depending upon its precise transcriptome, its proteome, its structure and function, then at any given point in time, the endothelium would display a rich color palette. Certainly between arteries and veins and capillaries between different vascular beds, if not between neighboring endothelial cells in the same blood vessel wall. And if we were then to roll the film, insofar as that environment changes over time, these colors may blink on and off like lights on a Christmas tree. Now, what I've described so far depicts the endothelium as a blank slate, kind of marching blindly and slavishly to the tune of the microenvironment. However, as I'll show in the next couple of slides, the story is a little bit more complicated than this. So one way to think about endothelial heterogeneity and its mechanisms is to look at sort of this time-honored dichotomy in the behavioral biosciences of nurture versus nature. So you can consider the following thought experiment. Take two phenotypically distinct endothelial cells from the body, from their intact tissue. It could be from the blood-brain barrier versus the capillary in the heart, but for purposes of illustration, we'll consider two endothelial cells that are virtually side by side in the same blood vessel. Remove them from their native environment and culture them under identical conditions. If these differences in phenotype are dependent upon the microenvironment of their native tissue, then these phenotypes will drift towards one another and ultimately reach identity. An alternative is that these differences are somehow epigenetically fixed and forever retained under culture conditions. Now, it turns out that there's a lot of literature that have indirectly looked at this, and the answer, which shouldn't be surprising to anybody, lies somewhere in between. That is, while some properties drift or wash out in vitro, others are mitotically stable. And the distance between these two color codes is inversely proportional to the plasticity of the endothelium. A really interesting hypothesis, which has found some evidence in the literature, is that disease and aging is associated with divergence of these colors. In other words, less plasticity and more lock-in. Not a good thing. Now, while the nurture side of the equation is readily explained by imminently reversible, environmentally responsive signal transduction pathways, the nature side is a little bit more complicated, but presumably involves some combination of a change in the histone code and DNA methylation. Which mechanism is operative is important because as far as the environment is controlling heterogeneity, one might choose to target that environment, whereas epigenetics, one has to target the cell itself. Well, I asked at the beginning how we look at the endothelium, and I view the field as being separated into two extremes, the lumpers and the splitters. So the lumpers don't really care about the color palette. They want to know what the default phenotype of the endothelial cell is. Tell me what is the difference between an endothelial cell and a smooth muscle cell, or between an endothelial cell and a cardiomyocyte. The splitters, and I'm an avowed splitter, are much more interested in the color palette, and the more colors, the more interesting. How is the color palette developed? How is it maintained? And how does it go wrong in disease? I'm not suggesting that we form a discipline for each phenotype of the endothelial cell. On the contrary, the endothelium, in my opinion, should be viewed as one large, gigantic mosaic, whose colors are not randomly distributed, but rather conform to certain patterns that are influenced and shaped by evolutionary, by developmental, and by environmental forces. And these patterns, and this is a very important point to make, these patterns pay no attention to artificially imposed boundaries between organs. They carve their own paths. So I want to finish with just two of many possible examples. I want to take you back to the example of preeclampsia. This disease starts in the placenta, but it spills out into the maternal circulation where it affects the brain, the kidney, and the liver. So what is it about, and the microvessels in these organs, what is it about the color palette that ties these various pathologies together? It turns out to be a phenotype, or color, if you will, of the endothelial cell that makes it particularly vulnerable to circulating anti-angiogenic factors. And that is those endothelial cells that form fenestrae and are dependent upon VEGF for signaling in the choroid plexus, in the sinusoids of the liver, as well as the glomeruli of the kidney. The second example is Goodpasture's disease, which is a rare autoimmune disorder that attacks the lung and the kidney, leading to pulmonary hemorrhage and renal failure. The common denominator here happens to be a component, or in this case, antigen of the basement membrane that is released by endothelial cells only in the lung and the kidney. So it's not good enough for the pulmonary folks to say, well, I'll take care of my vascular bed and you renal folks take care of your vascular bed. I think it's important as we move forward to be able to navigate much greater swaths of territory of microvessels, either independently or preferably in teams, and to be able to decode the color palette and identify novel patterns in the color palette that might be leveraged for therapeutic gain. So I'll end on that note. Thank you for your attention. Happy to take any questions.
Gary Meininger from University of Missouri. I wonder if you might comment on endothelial microparticles and their potential for clinical use, and also gender differences in the endothelium. Do you have any comments about that?
Well,
Have a talk about
yeah, we have two talks from now. We have a talk on gender differences, so I'll leave that to the speaker, and I wouldn't be able to answer that with any justice. In terms of microparticles, absolutely, and I should have included that in potential novel diagnostic technologies. And I think those technologies with phenotyping microparticles with novel sort of FACS technology are advancing. So absolutely.
John Lederer, University of Maryland. Question, lumpers, splitters. Isn't there a third possibility? I would call it that, local control. Suppose each of the endothelial cells really sort of starts off the same, more or less, and is controlled locally by its environment. Would be good for, say, neurovascular coupling, that sort of thing.
Yeah, so I mentioned the two extremes, and there are obviously some middle ground, and that middle ground sometimes involves patches of color.
But isn't this conceptually different? It's not programmed, it can be adaptive. So if you have a pathologic environment, you can actually change the endothelial cell and its function under local control.
Right, but that color palette doesn't depend upon the relative roles of the environment regulating it and the epigenetics.
Ariel Gomez, Charlottesville. I really enjoy your talk. Tom Davis alerted me to one of your wonderful papers, a review paper. I can't remember the title at the moment, but in that paper you ask questions about how versus why. And we found using a different methods that in the kidney vasculature, the renin cells, as well as the smooth muscle cells, actually are arranged in a mosaic pattern using chimeric mice as well as the confetti mice, and show this multiclonal as well as a mosaic distribution. So, given that you are interested in asking why and with the purposes of the discussion, I wonder if you care to speculate how this might be determined even at the developmental level. I mean, what would be the purpose of this? You know, they are arranged like one side by side, and they are multiclonal in that regard. I don't know the answer.
Yeah, so I'm not familiar with that particular phenotype. Maybe we could talk after.
Well, it's not published yet, but...
Okay, yeah. So we'll talk more about it, and I might be able to give some why answers, but I'm not familiar enough with it.
Okay.
Beautiful talk, Bill. Bill Chillian, Northeast Ohio Medical University. Just one question. What would you do with the lymphatic endothelium?
Oh, yeah, so I knew that question was going to come up. And I didn't have enough room on that first slide when I talked about classification, where I really should on the left-hand side before I even got into pulmonary versus systemic circulation, have that open-ended lymphatics coming off. Absolutely critical component of the cardiovascular system, sort of the poor second cousin that gets ignored and shouldn't be. So thank you for pointing that out.
Marilyn Cipolla from Vermont. Really beautiful talk, really great opening session. I really appreciate you bringing up preeclampsia. And I want that theme to run throughout here because I think it's an incredibly important one that we're, disease that we're just, kind of recognizing in terms of the cardiovascular risk, right? I just want to point out, however, that it's not all about the placenta. And it's not, many women who have preeclampsia do not have placental ischemia. And I think the thinking on this in terms of the spiral artery modeling and the lack of it has really pigeonholed our thinking. And it has really gotten us away from really understanding the flip side, which is the maternal endothelium, which is dysfunctional. It's not just about SFLT and VEGF. There are many women, especially who have eclampsia at term, who don't have placental disease. And so as we move forward and talk about preeclampsia more throughout this workshop, I just want everybody to remember that while there are women with placental ischemia that you're talking about, there's a large proportion of women who have preeclampsia without placental disease. And if we're going to ever solve this disease, we need to start thinking about it in terms of the maternal endothelium as well as on the fetal side. So,
Thank you for pointing that out.
It's a really beautiful talk, Bill. Hong Wang from Temple University School of Medicine, Philadelphia. You touched two fundamental points about the plasticity of the endothelium in the microcirculation, brain versus the heart. So we understand that different microenvironment surrounding cells contribute. And the second point you mentioned is the epigenetic difference. Do you have any thoughts about what determines the difference of the epigenetic environment in different organs that may contribute to the plasticity of the endothelium functional structure?
Well, I think most of the epigenetic programming occurs during development. Although there's a lot of exciting recent data that implicate a role for epigenetics in physiology and pathophysiology in the adult. For example, in atherosclerosis, some great work by Peter Davies.
Tissue difference Anything?
The tissue differences, it's very, so we know about the importance of epigenetics, and we have understanding of mechanisms underlying epigenetic differences between lineages, endothelial cells and non-endothelial cells. The extent to which epigenetics contributes to endothelial heterogeneity per se, which is your question, for example, brain versus heart, is a completely open area that's ripe for study. I don't know, not familiar with any study that has actually addressed that question and define mechanisms that are responsible for that. But by definition, they exist. We know that they exist because a lot of these cells have memory in culture.
Vesna Garovic, Mayo Clinic. Actually, I want to extend a little bit more on preeclampsia and placental dysregulation, because 10% of women with preeclampsia develop disease postpartum when placenta is long gone. So, you know, just extension of the previous commentator, Dr. Cipolla, that, endothelial dysfunction as a consequence of placental dysregulation is very important, but that maternal endothelium, maternal sick endothelium, especially as we see more and more women having a first pregnancy in their later age, when they may have more cardiovascular risk factors in general. We see an explosion of obesity and increase in BMI in pregnancy. So that maternal endothelium really should be focus of investigation as pivotal in causing this disease, especially in postpartum cases when placenta is long gone.
Right. So I think the important message here is it's not just about soluble endoglin and soluble platelets. It's not just about the acute syndrome, but also long-term effects in cardiovascular disease.
Which makes the study of maternal endothelium even more important.
Right. Thank you.